So, again, thank you very much. So, I guess you are, just as a reminder, most of you are aware of this, but just as a reminder. So the genome encodes genes which contains the instructions for making proteins, and proteins are in turn the machines of life that are involved in all functions, that the extraordinary number of functions that occur in our bodies. And variations in proteins, and in turn encoded in the genes, determine also variations in disease susceptibility, longevity, and aging. And so in a sense, the genome is the digital code of life that, to a very large degree, I would argue, determines our traits, determines our characteristics, including in the context of aging and longevity. And we also know, particularly from studies in model work, but also from some information from humans, that even though the genome is very big, it's 3 billion base pairs, very tiny, very subtle changes in the genome can have a major impact on aging, on longevity, on age-related diseases. So this is an aspect that's always fascinated me about biology, genomics, and aging. And so what we're trying to do is, the goal of our work is to help decipher the human genome to understand how it regulates aging and longevity. And I guess we do basic research, though my ultimate goal, or the reason I became interested in aging, was not just because I find it interesting from a purely scientific curiosity perspective, but I would like to intervene in aging. I guess the ultimate goal of what I do is to help develop technologies that extend life and preserve health. I think in particular in context of genomics, this is now being certainly much more easier thanks to massive advances in sequencing technology. So it's much cheaper now to sequence genomes than it used to be. So what you see here, so this is years, this is cost, and what you see here is Moore's law, which I'm sure most of you are familiar with, which is the declining costs of computing. And what you see here is cost of sequencing, which, as you can see, I guess it's leveling off a little bit now, but still it had these massive drops in the last few years. What it means is that while the human genome costs, I think, a couple of $1 billion to sequence, you can now sequence your own genome for about $2,000, maybe less, depending on quality. So that means that we have this great capacity to generate data. And in a sense, biology is becoming a mathematical problem, in the sense that it's much easier now to generate data than it is to analyze it. And this is, I think, has a lot of potential and applications in aging and longevity. I don't think it's too surprising that there are now a number of companies and labs focusing on these large-scale approaches to longevity and to aging, like Human Longevity Incorporated, the Institute of Systems Biology, which have, I think, called Wellness Project. Google have their company. It's not entirely clear that will focus on a genomics level, but I think it's quite likely. So with that in mind, I guess our lab, what we do is we do a combination of experiments and computational approaches, from particularly cell and molecular biology, and then we try to take advantage of these large amounts of data, some that we generate, others that we employ from public databases, essentially to gain insights on life, and on aging, and on longevity. So with that said, I'll tell you about just one particular direction that we'll be focusing, and that's in studying the genome of long-lived animals. I think on one hand there's a curiosity-driven approach to this, which is that, as I'm sure you're aware, different animals age at different paces. So, mice, for instance, mice and rats, no matter how well you take care of them, they will age about 20, 30 times faster than a human being. So there has to be these differences between species must be seated at the level of the genome. And on the other hand, of the spectrum, we have humans who can live over 100 years, and whales which can be estimated to live over 200 years. So if we can understand these species differences in aging and longevity, that could give us information on the underlying mechanisms, causal mechanisms of aging. And the rationale for this, for studies of long-lived species, is also that if we can understand the genes, the pathways that confer longevity and disease resistance in some organisms, these may have human applications. I will come back to that right in one of my last slides. So what we've been doing, we've been focusing on long-lived species. I guess one of the first that we focused on was the naked mole rat, which I'm sure many of you are aware of, this lovely animal. So the reason we're interested in naked mole rats is because, first of all, they're quite long-lived, so they can live over 30 years. They're, as far as we know, the longest-lived rodent. But most of all, because they're extremely cancer-resistant. So while about a third of the people in this room and 20 will have cancer in their lifespans, and well, most mice do die of cancer, naked mole rats, as far as we know, don't get cancer. It doesn't mean that they will never have cancer. But in about, I think it's been hundreds of animals studied in captivity, no cases of cancer have ever been found. So I think that's quite a remarkable trait. I'm not aware. I think there's other mole rats which also exhibit similar cancer resistance, but otherwise I'm not aware of another mammal with such cancer resistance. So back when I was in Boston as a postdoc, we put in a genome sequencing proposal to the NIH, which was unfortunately unsuccessful, but I think that fostered then genome sequencing efforts of these organisms. So it was sequenced by the BGI and more recently by the Broad Institute. And so one of the things we've recently done, and in collaboration with the Broad Institute, was we created this portal called the Naked Mole Rat Genome Resource. It essentially, it has all of the information from the genome. It has gene annotation, proteins. You can search for particular proteins that you're interested in. So the idea is that this will help facilitate analysis and further studies of this fascinating disease-resistant animals. So this is now available on this website, and the paper is now, I think it's online already in bioinformatics. We've also done, I don't have time to go into great details, but we've also looked at particular genes that could be responsible for naked mole rat specific adaptations. We found, so at the level of P53, we found a few genes of potential interest that, for instance, the BGI analysis missed. One of them was in P53, so what you see here is something called proline-rich domains, which are these little boxes here. So you have naked mole rat, guinea pig, mouse rat, and human. And as you can see, there seems to be more of this in human than in human and naked mole rat than in other species. And there's some evidence that this could be related to apoptotic responses and might be related to cancer, though I think there's still a little bit speculative, but it is possible that this indicates convergent evolution at the level of particular domains in P53 in naked mole rat and humans. I mean, we found other results that I won't go into detail in BRCA1, and interesting also in the hyaluron receptors. So those of you who are familiar with naked mole rat biology know that Varga showed that hyaluron pathways is important in the cancer resistance of these animals. And so I think it's interesting to see that there's differences in its receptors. And I mean, actually this paper is also in collaboration with Vera and Andrei Salanov. Now the other long-lived animal we've been focusing on is the bowhead whale. And the reason we're interested in these animals is, first of all, they live a very long time. So they've been estimated using indirect biochemical methods that I can go into detail later if you wish. But they've been estimated to live over 200 years. And the other interesting aspect about it is that, so they're quite big. So the second biggest animal on Earth after the blue whale. And if you think about cancer as emerging from one rogue cell, then these animals, because they have about 1,000 times more cells than human beings, then if the chances of developing cancer from one cell becoming cancer are the same for humans and bowheads, then they would have a lot of cancer. Only they don't. I mean, they can get cancer, but they don't have more cancer than human beings. If anything, they have less. So they must have some mechanism not just to live long, but to protect against diseases like cancer. So what we've done is we've sequenced the genome. Again, I won't go into a lot of methodological details, but we generate about 150x coverage. So if a genome is 3 gigabytes, it's about 450 giga of data. I mean, then what you do, so with the sequence genomes, nowadays with next generation platforms, you have a lot of small fragments, and then it goes into computational cluster that put everything together. And then we also generated RNA-seq data from multiple species, and that allows you to figure out where the genes are, where protein coding genes are, and also what non-coding RNAs are. So we generated a genome. It's about 2.3 giga, which is similar to other whale genomes, and we have an annotation with about 23,000 proteins, which again is in line with other genomes. And then what we're doing is, excuse me. What I've been doing is trying to find genes that could be interesting, genes that could be related to the bowhead-specific adaptation. So comparing essentially, so this is the bowhead, comparing the bowhead whale genome with a minke whale, with a killer whale, with dolphin as well, with a cow, etc., to try to identify changes that are specific to the bowhead, try to identify genes that may be under selection in the bowhead, so more likely to be related to species adaptations. I don't have time to go into great details, but I'll give you a quick overview. So the first point is that a relatively small number of candidate genes under selection in the bowhead. That's not too surprising because the bowhead and minke, so the bowhead and minke are two closest relatives that we're using. Their genomes are actually quite similar, so you find a lot of very similar proteins. So you find relatively few genes and proteins with large number of differences between them. So there was a relatively small number of candidates. There were some potentially interesting bowhead-specific mutations in cancer-related genes, particularly genes related to cell cycle and DNA damage responses. There were also gene duplications in genes related to DNA repair, cancer, cell cycle, and shortened lifespan. So we didn't find these signatures in any gene associated with life extension in rodents, for instance, but we did find some bowhead-specific mutations in genes related to shortened lifespan and accelerated aging in rodents. So that's most what we found that could be related to disease resistance and longevity. And then I guess there were other findings related to sensory perception, dietary adaptations, immune responses, etc. The last thing I would say is also that, so I get asked this a lot. We did this analysis for the bowhead and we did an analysis for the naked molar, and there was very, very little overlap. Actually, there was practically no overlap between, I think there's like one gene that might be overlapping between the two of them. So what that suggests is that the mechanisms that, well, not the mechanisms, the genes that could be related to extended longevity and disease resistance in the bowhead are not necessarily those that are related in a naked molar, although they could still be similar pathways. So I think that's about it. So we're also working on a bowhead whale genome research. It's not fully operational yet, but we hope to have it soon. The manuscript is currently submitted. Fingers crossed it will be out in a not too distant future. The last thing I will mention is that, as I mentioned earlier, I'm interested in gaining knowledge that can be applied to preserve human health and extend lifespan. So what is the potential for translating these findings to human beings? Well, my rationale is that this is a new paradigm in using these long-lived and disease-resistant organisms to, contrary to what you would find in mice and rats, we're trying to identify processes and genes that protect against disease, rather than genes that are associated or they cause disease like you would do normally in mice and rats, etc., in short-lived organisms. So that's the rationale, and certainly with the dropping cost of sequencing, we can expect many more genomes of long-lived species, of short-lived species, and of exceptionally long-lived humans to be sequenced. And I think we can expect exponential progress in understanding of the genetic underpinnings of aging. I don't think translating findings to humans is particularly straightforward, however. Even if you find one gene that is related to, say, DNA repair in the naked molar in a bowhead, that is probably related to its phenotypes, is not particularly easy. I mean, we cannot just take the gene and put it in human beings. So I do think that is a barrier. As you're probably aware, there's a number of human genetic diseases that result from loss of function that we cannot cure, simply because it is, from a technical perspective, difficult to even if it's a normal protein, to just restore its function in a time and tissue-specific fashion. So I do think that that is an issue, even though I think that our understanding will exponentially increase in the next few years. I mean, I have a review coming out in Aubrey's journal, Rejuvenation Research, which is now available online, that goes into this in more detail. So I think that's it. Thank you very much. I should acknowledge, particularly for the work on, the work on the naked molar was led by FP7 and also by the LSM Medical Foundation. And the work on the bowhead was paid by the Life Extension Foundation and the Thistledown Foundation. And so these are the people who did the work. So Thomas, Michael were the main people involved in Thomas and Michael were the main people involved in the bowhead and the naked molar projects. So thank you very much for your time and attention.
Most liked in your interesting lecture was the last sentence, because it said exactly what I think, that it's not easy to translate findings on animals to humans. There is an extraordinary example, which I might, if I have time to explain in my talk, is calorie restriction. I don't know if you know the story. Calorie restriction is the only trick which worked to increase life expectancy. And from that moment, I predicted it will never work for humans, on historic reasons. And recently it was proved on monkeys at the National Institute of Aging. They become healthier, stronger, but they still die at the same age as if they wouldn't be much stronger. So extrapolation in biological research, even more so if it touches medicine, is impossible. Otherwise, contact will work.
Well, thank you. I wouldn't go to say it's impossible. I certainly agree it's a big problem. I completely agree with you with caloric restriction. I would add, I agree with you, but I would add that I think part of the problem with caloric restriction is that most of the studies done in model organisms are done on particular laboratory strains. And it works, and what we know now is caloric restriction, for instance, in mice works for some strains, but doesn't work for others. Which, again, raises question about extrapolations to humans, as you were mentioning, and I completely agree.
It's mainly a philosophical question. It always comes up when what we call in French a polytechnicien speaks to a physician. You see, we see the variety, the same disease in two humans is not the same. You see, so generalizations are very, very difficult in biology and